All right, in this video we're going to look at the structure of the cell membrane. The cell membrane is made up primarily of phospholipids, which are a type of lipid, if you remember from the chemistry review. There's a phosphate head. The phosphate head is hydrophilic or water-loving, so it's going to be on the outside. It's attracted to water. The fatty acid tails are hydrophobic. They don't like water, so they're going to orient themselves so they're in the center. They're repelled by water. So this causes this phospholipid layer to be a bilayer where the heads are on the outsides and the tails are in the middle. You can see that in the picture here. So it serves as a cellular barrier or border. It defines the edge of the cell, but it also controls what can enter and leave the cell. Lipids can go through. Sugars can't go through. Water can't go through the membrane. Salts can't go through the membrane. Waste can't go through the membrane. Polar molecules in general cannot go through the membrane. Nonpolar things like lipids can. The cell structure must be more than lipids, though. In 1972, Singer and Nicholson proposed a membrane proteins are inserted into the phospholipid bilayer in their fluid mosaic model of the cell. So it's fluid, then that it changes, it moves, things are not set in place. It's a mosaic because things appear at least to be randomly scattered through the cell membrane. So you can see some of the different structures here. You can see the phospholipids, cholesterol, proteins, carbohydrates, lipoproteins, glycoproteins, and so on. So here you can see how it's all arranged. So the carbohydrates, or what are usually attached to a protein, so it'd be a glyco or a sugar, glycoproteins, things like that. They're important for cell-to-cell recognition. They allow cells to distinguish one from another. They also allow things like our immune system to recognize our cells and not attack them, and recognize foreign cells and attack them. There are antigens. These are the markers that the immune system recognizes and things like that. They're important in organ and tissue development. And like I said, they're the basis for rejection of foreign cells by our immune system. The cell membrane defines the cell. It separates the living cell from its surroundings, from, in many cases, the aqueous environment or water environment. It's a thin barrier. It's about 8 nanometers thick. And like we said earlier, it controls the traffic in and out of the cell. It regulates what goes in and out of the cell. It's a semi-permeable membrane. Some things can cross it, other things can't. Hydrophobic, nonpolar things go through easily through the phospholipid layer. Hydrophilic, polar things need help to get across.